on to the next talk, which is by Vanessa Couldridge, which is on predator avoidance.
Thank you. So good morning, everyone. And today I'll be talking about some of the work that we've done on Bullacris unicolor, which is a species of bladder grasshopper. So to start with, just a brief introduction on bladder grasshoppers and what they are. So bladder grasshopper is the common name given to a group of grasshoppers belonging to the family Pneumoridae, which is a small group consisting of 14 species. They are found only in Africa, nowhere else in the world. And specifically, they are found in southern Africa, the coastal regions of southern Africa. So their distribution extends from southern Namibia all the way around the coast of South Africa. And then we have two species that have distributions that extend into East Africa as far as Uganda. So they are sexually dimorphic. Males and females look different to each other, as you can see in the picture there. So this is a male, and that's a female of the same species. Only males have the inflated abdominal bladder, which is where the common name comes from. And the purpose of the bladder is to amplify sounds. So it allows them to produce a very loud call at night, which can transmit for distances of up to 2 kilometers. So that's the distance at which they can hear each other. All species are cryptically camouflaged, which makes them difficult to detect, which also makes them difficult to work on, and they are active at night. All right, so people have long been fascinated by bladder grasshoppers and the sounds that they make. Charles Darwin even mentioned them in his book, The Descent of Man. So the way in which the males make the call is it's a foul and scraper mechanism, so there's ridges on the back leg that they rub against another set of ridges on the sides of their abdomen. And I'll just play that for you so you can see how that works. Okay, so the purpose of the sound is to attract mates. Males will call to attract females. Receptive females will respond acoustically to the male call. And then males will try to track the female. So only the males have wings. They're able to fly, so they fly around looking for females. The females don't have flight wings, so they're on the bushes, and the males try to track them. So I'll just play the acoustic duet between the male and the female. This is a different species, so I apologize for the quality of the video. It's filmed at night, but you can at least hear the duet. Andy found her. All right, so many animals that use sound to communicate will call preferentially at certain times of the day or night, and there are many different factors that can influence when animals prefer to signal. So we know that abiotic factors influence signaling activity. So for example, temperature, preferring to call at warmer temperatures, or when conditions are less windy. Acoustic competitors can also influence signaling activity, so they may try to avoid competitors. Those competitors may be of the same species, or they may be of different species, so particularly species that call in the same frequency bandwidth. They may also time their calls to try to avoid predators, so preferring to be active when predators are less active. And increasingly, we are seeing the effects of anthropogenic factors. So this includes noise pollution and also artificial light at night. So we use Bullacris unicolor as our study species to look at signaling activity patterns. Bullacris unicolor is a species that occurs along the west coast of South Africa, so in the Western Cape, and also in the Northern Cape, so you find it this region. So its distribution spans 2 vegetation biomes. We have the fynbos in the south, which is a shrubland, and then the succulent karoo further north, which is a semi-desert region. So we had noticed previously that males from these two different regions differ in when they signal during the night, and in particular males further north, in the northern part of the distribution, will signal much later in the night. So you typically hear them in the early hours of the morning, just before sunrise, whereas further south they will start calling after about 10 p.m. So we wanted to find out why is this? Why do males in different parts of the geographic distribution signal at different times during the night? So we conducted passive acoustic monitoring, which we did during the spring season, which is the peak seasonal activity of the grasshoppers. We had 6 sites, 3 sites in the northern part of the distribution in the Northern Cape, and 3 sites in the Western Cape representing the southern part of the distribution. So we put up recorders, we used the wildlife acoustics recorders, and we recorded from sunset to sunrise continuously, so during the night, and we did this for 3-week periods in the different locations. Alongside this, we also continuously logged weather variables, including temperature, humidity, and wind speed. So we did acoustic recording, and we also did ultrasonic recording. The reason we did ultrasonic recording is because we wanted to detect the presence of bats, which are believed to be the major nocturnal predators of the grasshoppers. All right, so from our recordings, we looked at calling activity, and we were able to calculate call rates. So we measured call rates as the number of calls per 30 minutes time interval. So we did this for our study species unicolor, and we also did this for any other bladder grasshopper species that we detected on our recordings. So just to play those for you. So that's unicolor again. And then in the southern part of the distribution, we found 2 coexisting species. So the first one is a Bullacris. Okay, so it's a very long call. And then we've got also Bullacris. Very short call. Okay, so that's just a spectrogram in the middle there, showing all 3 species calling at the same time in the same area. So it was quite easy to distinguish the different species. Bullacris obliqua has this very long call, which is easy to pick out. And then the 2 species with the shorter calls, Bullacris calls at a higher frequency than unicolor does. So it was quite easy to distinguish them from our recordings. So just what they sound like in the field. Okay, so a mixture of 3 different species there. All right, so from our recordings, we detected 9 bat species in total for the study, which varied from between 5 to 8 different species of bats per site. So we did find mostly the same species that occurred in the different locations, although the relative abundances did vary. So in our northern sites, our recordings were dominated by the Egyptian free-tailed bat, but in the southern locations, our recordings were dominated by the Cape serotine bat. All right, then if we look at our call rates, so what you can see at the northern sites is that there are distinct peaks of activity. So here we have the bats in the yellowy orangey color, and the grasshoppers in the blue color, and you can see there are very distinct peaks of activity that do not overlap. So the bats are active earlier in the night. That's when we detected most of the bat vocalizations, and the bats tend to taper off later in the night, and that's when the grasshoppers are active. So we see grasshoppers calling much later in the night, and they will continue to call until dawn. So this suggests that the grasshoppers are avoiding the bats. If we look at our southern locations, we see a different pattern. So here we see everything kind of calling together. It's a lot more messy. It's difficult to look at patterns. So what we do notice is that the bats are active for much longer periods. So again, the bats are in yellow, and we see that they are active throughout the night. If we look at our grasshoppers, we see unicolor, which is shown in blue there, is active much earlier in the night in the southern locations, which we already knew, but it is active at the same time that the bats are active. And then our coexisting species, so we have Physomacris in white. Physomacris, there is some temporal partitioning between the different bladder grasshopper species. So Physomacris tends to call a little bit earlier in the night than unicolor does, although there is some overlap, and Bullacris occurred in very low numbers, so we excluded them from our statistical analysis, just to point out that bladder grasshoppers are considered a group of conservation importance. So a lot of the species are observed to be in decline. Bullacris obliqua is one of those species that you don't encounter all that often, so we did pick it up, but not in enough numbers to do any kind of analysis with that. In terms of our weather data, when you looked at correlations with temperature, we found that temperature was negatively correlated with call rates at the northern locations, but positively correlated with temperature at the southern locations. And the reason for this has to do with when the grasshoppers are active. So because they are calling much later in the night at the northern locations, it means that temperatures are very low at that time of night. And that's why we see these negative correlations. Although they would prefer to call on warmer nights in general, they are calling in the coldest part of the warmest night. So we see these quite complex relationships with temperature. For relative humidity and wind speed, both of these had a generally negative influence on call rate. So the grasshoppers preferred to call when conditions were less humid and there was less windy conditions. All right, so to conclude, geographic variation in predator activity has likely led to interpopulation divergence in dial signaling behavior. Weather conditions and interspecific competition did also affect timing of signals, but they could not account for the delayed signaling that we observed at the northern sites. And we know that signal timing appears to be fixed rather than a plastic response, which would have implications for species integrity. So if you take the animals out of the field and you put them into a lab, so you take a grasshopper from the Northern Cape, you bring it to Cape Town and put it in the lab, it will continue to call at 5 o'clock in the morning. So it doesn't adjust based on local conditions. So it suggests that there might be some kind of genetic determination to the time at when they call, in which case there may be divergence going on between these populations, and previous work has shown that there is some genetic divergence between them. Okay, thank you.
All right, thank you so much, Vanessa, for a great presentation. We have time for one question. Yes, Eddie.
Thank you. I'll be quick. It's more of a comment. Would be super interesting to accompany your study with a diet analysis of those bats, to see how many
Yes.
proportions are actually of these species in their diet.
Yes, it would be very interesting to do that. We haven't done that kind of analysis. It makes sense that bats would be the major predator because and there's anecdotal evidence that we find the wings and the body parts under the bat roost, so we know the bats are eating them. It would be interesting to do a diet analysis.
Yeah, thank you. Great study, thanks.
Thank you.
All right, great. Thank you so much, Vanessa.